model of the molecule of carboxypeptidase, a sequence of amino acids folded together to give this three-dimensional structure. And it's this three-dimensional structure which determines its function as an enzyme. But this, the culmination of years of precise X-ray analysis of the enzyme, is the construction of an electron density map, a direct representation of the structure of the crystal, and the matching of these densities with the known shape of amino acids, research performed principally by this man.
I'm just completing the building of a small molecule which binds to an enzyme. This small molecule you can see in a reflection in the mirror inside some contours on plastic sheets beyond a lightly silvered mirror. This molecule is about 1/8 the size of the enzyme carboxypeptidase and blocks the activity by binding in the region of the active site. We're going to look in more detail at that interaction later. The enzyme itself, carboxypeptidase, digests protein in your food. So now let's take a look at how this enzyme operates. An amino acid has an amino and a carboxyl group. Peptide chains are formed as a carboxyl group joins to an amino group with loss of water to form a peptide or amide bond. A long sequence of amino acids can be formed, but there will always be an amino group at one end, the N-terminus, and a carboxyl group at the other, the C-terminus. Now, the enzyme carboxypeptidase selectively removes the C-terminal amino acid by cleaving the peptide bond here, preferably if there is a large uncharged side chain in this C-terminal amino acid, R group. This enzyme will then cleave the next C-terminal amino acid and so on down the peptide chain of the substrate.
But how can an enzyme work so precisely? To understand this, we need to look inside the enzyme and examine its three-dimensional structure. But how can we observe something as small as a molecule? The secret lies here in this simple demonstration. These ripples in water are analogous to a light wave, the dark being the peaks, the light the troughs. Now when two closely adjacent slits are placed in the wave path, two new wave fronts are produced which affect one another. They interfere. And here in the light areas, where the waves are out of phase, out of step, their amplitudes have cancelled one another out. Here in the dark areas, where they are in phase, their amplitudes have added together and so enhanced one another. And it's the same with light. When a laser beam shines through two such slits, interference or diffraction patterns are also produced. The intensity and positioning of these bands is related to the relative sizes and positions of the slits. The diffraction patterns are only produced when the distance between the slits is comparable to the wavelength of the light source. In a molecule, interatomic distances are thousands of times less than the wavelength of light, and so we need to use X-rays where the wavelength is comparable to these distances. So when X-rays are passed through a crystal, we would expect a diffraction pattern to be produced. In this case, a Polaroid film is being used to detect the pattern, the X-rays exposing the film. It's this pattern which tells us the relative positions of atoms in the crystal. But there is a problem in deciphering this information. Let's turn to a laser experiment to explain this. Dr. George Harburn, Cardiff University.
One of the nice things about lasers is that their wavelength is so much longer than X-rays, so we can look at the diffraction patterns of much larger objects. I can look at the diffraction pattern on a screen here, and I'll just focus it up. Just as with X-rays, where the diffraction pattern is related to the atomic structure of the crystal, so this diffraction pattern is related to its object. It's Mickey Mouse. Another nice thing about light is that it can be focused. This means we can go straight from a diffraction pattern to an image, which is essentially a second diffraction process. And this is exactly what we'd like to do in the X-ray case. Unfortunately, there's no way of focusing X-rays and so we have to go through an intermediate procedure of recording the X-ray diffraction pattern. When we do this, some information is lost. It's exactly the same with laser light. If we take a photograph of an optical diffraction pattern and use it as a diffracting object, Mickey Mouse again, then the diffraction pattern of that ought to be an image of Mickey Mouse, but in fact it's quite meaningless.
So once a diffraction pattern has been recorded, information is missing, and this is the phase. Let's look at some simple physics. Consider one atom only. When X-rays are directed onto it, they are scattered in all directions. One of these waves is illustrated here as a sine wave. The amplitude is the maximum excursion above this line. An atom with more electrons scatters with a greater amplitude, so we need to measure amplitudes in order to study atoms. But we can't do this directly. By measuring intensity of these diffraction spots, we're actually measuring the square of the amplitude, but it lacks phase reference. We don't know the relative shift along the horizontal line of the two waves. These two waves are in phase. These two waves are out of phase, since the amplitudes do not coincide. When we have two atoms, we can have the scattered waves in phase, giving an intense spot, or out of phase, giving a reduced or even zero intensity of the diffraction spot. But without the phase information, we can't reconstruct an image of the atomic layout in the crystal. But crystals are far more complex than two atoms. They are composed of regular repeating identical unit cells. Each unit cell shares its atoms with other cells. In the case of salt, the unit cell has 4 atoms of sodium and 4 of chlorine, and it's the interfering of the scattered waves which produces a diffraction pattern. But what's the effect of the repeat within the crystal?
If we use a regular arrangement of dots, holes, with a laser and look at their diffraction pattern, then we'd expect to build up the sort of diffraction pattern that you'd get with X-rays from a crystal. If we use just one hole, then the diffraction pattern on the screen here is in fact very faint, and we have to look at it with the lights off. With just one hole, the diffraction pattern is broad and diffuse. If we increase the number of holes, then we produce diffraction maxima. If we have a row of holes, the maxima are sharp in one direction. If we use a 5 by 5 array, the maxima are sharp in both directions. With a 10 by 10 array, the maxima become still sharper.
Proteins like carboxypeptidase can be crystallized. A crystal like this has taken over 3 months to grow. The know-how, however, took several years, but it's worth it. X-ray crystallography of proteins depends on it. In the same way that arrays of dots increase the diffraction maxima with lasers, so the regular repeating units in the crystal of the protein produce strong X-ray diffraction peaks. By rotating the crystal, a complete diffraction picture of the molecule is built up.
Here we have a diffraction photograph of a crystal of carboxypeptidase A. The unit cell contains 2 molecules and therefore 5,000 atoms of carbon, nitrogen, and oxygen. Each of these 5,000 atoms contributes to each of these diffraction maxima. But these various maxima are not necessarily in phase. In fact, the phase information has been lost in the experiment, and with this loss of phase, we are unable to solve the structure.
The first protein to be crystallized and have its structure interpreted was myoglobin, and the phase problem was a stumbling block here, as the late Professor Sir Lawrence Bragg recalls.
This picture is very similar to the one which Puritz brought to you to show you. Can you remember your reaction when he brought that to you?
Yes. The thrill of seeing so much information on it. Because what we realized was that if only we could interpret it, it was there.
What's there?
There was sufficient information in all those spots
Yes.
to give an answer, if only we were clever enough to find it out. The crystal was telling us something if we could only interpret it.
So by measuring only intensities, phase information was missing, and the diffraction pattern couldn't be interpreted. In fact, it was Max Perutz who unraveled the secret using a simple but elegant technique.
The key to finding these phases lies in the use of heavy atoms in the protein structure. Here is a diffraction photograph of two different crystals superimposed slightly out of register. The lower diffraction pattern is from the pure protein crystal, and the upper from a protein crystal which has lead atoms added to it. There are intensity changes. They are particularly clear along this line here. From these intensity changes, we can establish the relative phases. Let's see how it works in theory. Suppose we know the protein structure, which we represent here as a set of 4 atoms. The scattering relative to the origin is at a maximum in the neighborhood of these 4 atoms. If we add a heavy atom here, the phase is displaced to the left. And if we add a heavy atom here, the phase is displaced to the right. And now we can refer all phases to the heavy atom, which can easily be located in the crystal. In practice, we can obtain the phase from the position of the heavy atom and the intensity changes produced by the heavy atom. Now, armed with these phases and the amplitudes, we can then compute a 3-dimensional electron density map, which is displayed as a series of contours of increasing density. The computer displays the map as a series of transparent parallel sheets on which the contours have been drawn, giving a 3-dimensional picture of the electron density of various regions of the molecule. We can trace most of the polypeptide chain in the map, and we can see regions where the density is very large, corresponding to groups with large numbers of electrons. Here, for example, is the disulfide bond. And over here, the densest region of all is the zinc atom. This atom is absolutely necessary for the activity of the enzyme, and that is sometimes called a cofactor or coenzyme. We can recognize some of the side chains. For example, here is a histidine residue beside the zinc. However, we are not able to be sure of the trace of the polypeptide chain, nor can we identify all of the side chains without a linear chemical sequence. And this has to be established by biochemical methods. Now, we can use a computer display to fit amino acids to particular regions of the 3-dimensional map. This tyrosine side chain, number 248 in sequence, can be rotated and fitted into the electron density. In this way, the polypeptide chain of 307 amino acids can be folded into its correct 3-dimensional configuration. This is a long alpha helix. You can see a pleated sheet structure, 8 strands twisted by the L configuration of the side chains. And this is the active site cavity showing the zinc atom. We now look at the active site in more detail. A good substrate would be cleaved rapidly, so we must use a product or an inhibitor in order to find the active site region. Moreover, we must establish whether this product or inhibitor is competitive with the substrate itself. We have looked at 2 substrates in some detail. One of these is glycyltyrosine, in which the arrow shows the point of cleavage. The other is an inhibitor closely related to glycyltyrosine. When we bind this inhibitor to the enzyme, we then take a new set of X-ray diffraction data, subtracting the electron density of the enzyme itself, and we display the result in these three-dimensional contours. Here, in the region of the active site, where previously we had only water molecules, we see new density, the benzene ring in the pocket of the enzyme here, the other benzene ring of the inhibitor here, and the carboxylate group of the inhibitor here. In addition to the new contours of the inhibitor, we see the contours of the arginine side chain which binds to the carboxylate group at this point. In this set of contours, which is immediately above the original set, we see negative contours here and the appearance of positive contours here. This is the movement of tyrosine 248 to become near the enzyme substrate complex.
So both arginine 145 and particularly tyrosine 248 move in response to the binding of the substrate molecule in the active site. This movement brings charge groups together to ensure catalysis, and it provides the final evidence for induced fit in enzymes. And now we can see the active site in perspective.
This is about 20% of the enzyme molecule carboxypeptidase A in the region of the active site cavity where substrates bind. Here is the zinc atom, which is a cofactor or coenzyme, absolutely necessary for activity. And here is the negatively charged glutamic acid 270 residue, which will attack the carbonyl carbon of the substrate. The binding site of the substrate is this arginine 145, and the proton donor is tyrosine 248. Its movement upon binding of the substrate is a rotation about a single bond. In movements of this type, the substrate is enfolded by the enzyme. One might say that the enzyme embraces the substrate. Now, let's take a glycyltyrosine molecule, put it in the active site, and then we'll see how the enzyme does its chemistry. To bind to the active site, the substrate molecule needs to have the right shape and the right charges. Carboxypeptidase specifically cleaves the C-terminal amino acid of a protein. Let's see how the mechanism for this actually works. The first step is for the protein to bind to the active site. 2 hydrogen bonds are made to the arginine group. Also, water is displaced from the zinc atom, which forms a bond to the carbonyl oxygen of the substrate. The so-called tyrosine shift then occurs, enabling the tyrosine to form 2 hydrogen bonds with the substrate. An electron pair from the glutamate then forms a covalent bond with the substrate's carbonyl carbon, forming a tetrahedral carbon intermediate. A proton is then transferred from the tyrosine to the peptide nitrogen, and this allows bond cleavage to occur with a resultant cancellation of 2 charges. The peptide bond now has been broken, but the 2 parts of the substrate have to be removed in order to free the enzyme for further catalysis. 1 water molecule adds to the zinc, another to the tyrosine anion. The hydrophobic environment causes the zinc water molecule to lose a proton. there is nucleophilic attack by the hydroxide ion. The glycine is now free of the enzyme. A water molecule binds to the zinc. The tyrosine is now free to leave, and the enzyme's activity is finally restored. This is the model of the inhibitor that I was building at the beginning of the program. It fits into the active site of carboxypeptidase A, excluding the binding of substrates and blocking the activity of the enzyme. This inhibitor prevents the enzyme from activity in regions where it would be dangerous, as in the pancreas, where it is generated. When the inhibitor is removed, the C-terminal glycine has actually been cleaved from the enzyme. So what we're looking at is the molecular evidence for competitive inhibition.